Okay, students, sorry for that. I hope things are working again and no issues now. So let's continue. This is not required. Then there is a gel analysis of nucleic acid, which resolves DNA fragments of different sizes according to the size, shape, and charge. So hope you have all joined back. There were around 50 plus students, and there's again 53 students with us. So all is good. Why somebody is not able to join? So in gel electrophoresis, you have a DNA restricted fragments. We cut them with restriction enzymes, and then we place them on our gel, and then we apply electric field from negative to positive. So our DNA will pass through these pores and will pass. The shorter will transfer more, and the longer will remain up. So this is how the visualization of DNA RNA samples looks like by separated by gel electrophoresis. So we have 5 clones here, and these are the markers on the side. So you can see, so we have various markers of sizes, and in between clone 1, clone 2, 3, 4 of different sizes of molecular DNA are being separated in these 5 zones. This is also now divided into 2 parts. So all is good, okay. So this DNA gel electrophoresis, remember there are 3 types mainly. There is a southern blotting, and there is a northern blotting, and there is a western blotting. Yeah, southern, northern, western. Southern is done for DNA fragments, northern is done for RNA fragments, and the western is done for proteins. So SNW DRP. Remember this short, SNW DRP. So S for DNA, northern for RNA, western for protein. So specific sequence that is gene present in my DNA sample. So you have your DNA, right? This is your DNA. And then you cleave with the help of restriction enzymes. This is your gel electrophoresis running. You have filter paper, and then you can have, with the help of capillary reactions, DNA will be transferred to your nitrocellulose membrane, and your all DNA will be membrane. Okay. Then we hybridize the specific DNA with RNA loop or DNA probe on the autogram. So we will see some specific DNA markers with the help of that. And then for the northern blotting, this was done for total DNA, but this will be done for the total RNA. Again, the same steps will be done in this case, but then we will hybridize with your DNA probe in this case also, and then you will see your of beta globin mRNA after 96 hours so dark, and after 48 hours is so less light, and here untreated, it's not being treated. So this is how you can see your mRNA with the help of northern blotting. Then comes your real-time PCR for gene expression analysis. We will go through about these electrophoresis in upcoming lectures on the 6th day. So this was today. I'm just telling you about little bit about of each reactions, how they are working, how they are supposed to work. So so that we are aware of how gel electrophoresis, northern, southern, western blottings are working, what is the main principle. But we will go in detail with the virtual lab of them, of each of them, soon also. So in future, in I think in springtime, in let's say April, these links will might work out. So pin them down so that you can do them in the future. Sorry, they were all not working as of now. Otherwise, I have to show you these. But anyhow, let's continue with the real-time PCR now. Sometimes students, even some professors, they get confused with the RT-PCR as a reverse transcription PCR also. We ask them what is the full form of RT-PCR. They say it's reverse transcription because somehow name is quite similar to that, but it's always remember it's a real-time PCR actually. So what happened? This is also done. If you remember with the COVID test, COVID-19 test, your real-time PCR was done for gene expression analysis. So this allows real-time monitoring of PCR amplifications products by the use of fluorescence. So in the first step, reverse transcription of RNA into complementary DNA is done. Then either quantitative or semi-quantitative measurement of qualitative PCR is done. It's quite highly sensitive and used in the research labs for clinical diagnostic labs. So here you can see in the first case, you have a halogen tungsten lamp and they have excitation filters in it. Then they goes through our sample with the help of emission filters, then intensifiers, and then after with the help of various prisms, you can see them on the CCD chip, your results. So this is the plotting of PCR in real time is done. So you have your cycle number 0, 1 like this. At the 0 cycle number it's 1, at the 1 it's 2, at the 2 it's 4 like this. It's keep multiplying. And then by the 30 you will have in 1 billion base pairs of DNA. So the same case here, amount of DNA being produced in huge amount in this curve, PCRs per cycle. So this is quite interesting, this part 1, amplification curve to determine real quantitative express samples. So you have sample A and sample B. So and we can keep seeing the fluorescence of each sample like this. So initially you have a baseline, then comes the log linear phase, then comes the steady phase. So at the first phase, the time it starts to raise the threshold level, till that time it's known as your CT values, CTB values of sample B and CTA value of sample A. This is your cycle numbers, yeah. This is your cycle numbers. And then you calculate the output with the relative fold change with E target about divided by E reference. So E target raised to power delta CP target control minus sample and delta CP reference control minus sample. So this is the formula to calculate it. So let's see how this technique works. So you prepare the reaction mixture first with 2x Stackman master mix of 25 microliter. Then prepare the reaction mixture forward primers with 5 microliter per sample. And then reverse primer 5 microliter per sample. Then you take the Tachman probe, that is a 5 microliter per sample. So this is your reaction mixture is being prepared at the moment. Then alucot mix into 96 well plate. Then you add your complementary DNA sample 10 microliter. Cover the plate with the microseal film. Your plate is ready for running under the machine, under your real-time PCR machine. So you select your samples where it will work, then you analyze the results. So here you will see many peaks, data files. So this will be students, whenever you will go into PCR lab, every lab has its own softwares, every lab its own way of doing it. So I can explain you 10 years old experiments how it is done. So it's always better wherever the lab you will join it, they will teach you according to the software being used in that lab. So don't worry much about the mathematical calculations behind it, how things are working, how the results will be done. Just keep remember that the basic principle, how you prepare your things, how the reaction mixture is being prepared with the reverse primer and your forward primers with Taq polymerase and all the reactions that we have added into that, so that our reaction could run smoothly. So don't worry how things are done. So just go through the main principle behind it. What is the main principle? These are the peaks that you will see mostly working in the RT-PCR, which is corresponding to your cycle numbers. And then according to that, then there will be formula within your software which will count, which will create this regression and calculate the results for you automatically. Everything, you don't have to do anything in that. So further, let me show you how it is done.
Such as disease caused by severe syndrome coronavirus 2. When a person is infected, the most common symptoms include fever, cough, and shortness of breath. To start a test, the samples can be collected by a nasopharyngeal swab or an oropharyngeal swab. For nasopharyngeal specimen, the swab is inserted in the nostril and gently moved forward into the nasopharynx for a specified period time to collect secretions that contain the virus. Once the swabbing is applied, the swab is placed immediately into sterile tube containing a viral transport medium. The standard method of coronavirus testing is polymerase chain reaction, PCR, which is a method that used widely in molecular biology to make millions to billions of copies of a specific DNA fragment rapidly. Coronaviruses contain an extraordinarily long single-stranded RNA genome. To detect these viruses with PCR, RNA molecules must be converted into their complementary DNA sequences by reverse transcriptase. Then the newly synthesized DNA can be amplified by standard PCR procedures. This approach is universally known as RT-PCR. To perform this method, basically viral RNA should be extracted. Several RNA purification kits are available for convenient, fast and effective isolation. To extract the viral RNA by using commercial kit, the sample is first added into a microcentrifuge tube. Then it is mixed with a lysis buffer. This buffer is highly denaturing, and usually consists of phenol and guanidine isothiocyanate. Also, RNase inhibitors are usually present in the lysis buffer to ensure isolation of intact viral RNA. Once the lysis buffer is added, the tube is mixed by pulse vortexing and incubated at room temperature. Then the virus is lysed under the highly denaturing conditions provided by the lysis buffer. Once the sample is lysed, a purification procedure is carried out by using a spin column. The sample is loaded onto the spin column. Then a centrifugation is performed. This procedure is a solid phase extraction method, in which the stationary phase consists of a silica matrix. Under optimal salt and pH conditions, RNA molecules are bind to the silica gel membrane, and at the same time, protein and other contaminants are not retained. After centrifugation, the spin column is placed into a clean collection tube, and the filtrate is discarded. Then a wash buffer is added. The column is put in a centrifuge again, and DNA polymerase. Finally, to complete this reaction mixture, the RNA template is added. The tube is mixed by pulse vortexing. Then the reaction mixture is loaded into a PCR plate, which generally contains 96 wells, allowing the analysis of several samples at the same time. Next, the plate is placed in a PCR machine, which is essentially a thermal cycler. Real-time RT-PCR is used for the detection of the new coronavirus 2019 by the amplification of target sequences in the RDRP gene, the E gene and the N gene. The choice of the target gene depends on the primers and the probe sequences. The first step in RT-PCR is reverse transcription. The first strand complementary DNA synthesis is primed with the PCR reverse primer, which hybridizes to a complementary part of the virus RNA genome. Reverse transcriptase then adds DNA nucleotides onto the 3' end of the primer, synthesizing DNA complementary of the viral RNA. The temperature and duration of this step depend on the primer, the target RNA, and the reverse transcriptase used. Next, an initial denaturation step is applied, causing denaturation of the RNA-DNA hybrids. This step is required for the activation of DNA polymerase, and simultaneously the inactivation of reverse transcriptase. PCR consists of a series of thermal cycles, with each cycle consisting of denaturation, annealing, and extension steps. Denaturation step consists of heating the reaction chamber to 95 degrees Celsius, and it is used for denaturation of the double-stranded DNA template. In the next step, the reaction temperature is lowered to 58 degrees Celsius, allowing annealing of the forward primer to its complementary part of the single-stranded DNA template. The annealing temperature relies directly on length and composition of the primers. In the extension step, the DNA polymerase synthesizes a new DNA strand complementary to the DNA template strand by adding free nucleotides from the reaction mixture, the 5' to 3' direction. The temperature at this step depends on the DNA polymerase used. After the first cycle, the double-stranded DNA target is obtained. Then, the denaturation of this double-stranded DNA is performed, yielding 2 single-stranded DNA molecules. In the next step, the reaction temperature is lowered, allowing annealing of the primers to each of the single-stranded DNA templates, and annealing of the Tachman probe to its complementary part of the target DNA. Tachman probe consists of a fluorophore covalently attached to the 5' end of the oligonucleotide probe. The fluorescence is emitted by the fluorophore when it is excited by the cycler's light source. Also, this probe consists of a quencher at the 3' end. The close proximity of the reporter to the quencher prevents detection of its fluorescence. In the extension step, DNA polymerase synthesizes new strands. When the polymerase reaches a Tachman probe, its endogenous 5' nuclease activity cleaves the probe, separating the dye from the quencher. With each cycle of PCR, more dye molecules are released, resulting in an increase in fluorescence intensity, proportional to the amount of amplicon synthesized. This method allows the estimation of the amount of a given sequence present in a sample. The number of double-stranded DNA pieces is doubled in each cycle. Therefore, PCR can be used to analyze extremely small amounts of sample. For the measurement of the fluorescence signal, a tungsten halogen lamp, an excitation filter, mirrors, lens, an emission filter, and a charge-coupled device CCD camera are used. Filtered light from the lamp is reflected off mirror, passes through a condensing lens, and is focused into the center of each well. Then fluorescent light emitted from the wells reflects off the mirror, passes through an emission filter, and is detected by the CCD camera. In each PCR cycle, light from excited fluorophore can be detected by the CCD camera, which converts the light that it captures into digital data. This method is known as real-time PCR, which allows the monitoring of the progress of the PCR reaction, known as real-time PCR, which allows the monitoring of the progress of the PCR reaction as it occurs in real time.
So, them. Then with the help of reverse transcribe to cDNA labeled with the fluorescent dye. So you have a green DNA and you have a red dye. So then you have mixture of both hybridized to DNA microarray leading to the of them and measures green and red fluorescence over each spot. So you have your complementary DNA hybridized to DNA for single gene, and then you have array of like 8,600 genes on this tube, on this plate, and A is for the green and red is for the gene expression after the serum addition, and this is the genes decreases in the green spots. So that's how you get green, yellow, and red spots at the end. So each spots contain many copies of unlabeled DNA copies, and the black ones are where we don't have any corresponding mRNA present. So labeled cDNA hybridizes to spare where their sequence match. So red and yellow will be green, and identical expression both in both samples. This is the bioinformatic analysis in silico that can be applied to investigate common functions of clustered genes. So let us show you how it is done. The last reaction, last technique.
This animation will demonstrate how DNA microarray experiments are performed. DNA microarrays, sometimes called DNA chips, reveal the expression of thousands of genes on a solid surface, such as a microscope slide. In this example, we'll use yeast as a model system to illustrate one use of microarrays. One common use of microarrays is to determine which genes are activated and which are repressed when two populations of cells are compared. Every gene is measured simultaneously. As an example, we'll compare what happens to yeast genes when cells are grown in aerobic versus anaerobic conditions. The cells grow and adjust which genes need to be activated or repressed in order to survive. Now it is time to isolate the mRNA from both populations of cells. The cells are spun in a centrifuge. Now that the cells have gathered in pellets, we remove the liquid, but not the cells. Next, it is time to extract the mRNA from the cells. When we add the extraction buffer, the mRNA is released into the solution. Next, we remove the RNA and place it in a fresh tube. Now, let's make the cDNA from the mRNA. Here we see 3 of the many mRNA molecules from each tube of cells. Each mRNA is converted into red or green colored cDNA. When the colored cDNA is made, the mRNA degrades. Then we combine the red and green cDNA, mixing both colors into a single tube. At last, it's time to look at the DNA microarray. In our experiment, a microarray or DNA chip contains about 6,000 spots. Each spot is a different yeast coding sequence from a different gene. Let's choose 3 spots at random to follow in detail. Each spot is made of DNA that can base pair with its complementary cDNA. Here are partial sequences from each of the 3 spots we are observing. Now, let's incubate the mixed cDNA with the DNA chip. For the sake of our example, we'll zoom in and show that some of the labeled cDNA have bound to the DNA in the spots and formed base pairings. Here we see green and red cDNA bound to this spot. Only red cDNA is bound to this spot. And only green cDNA bound to this other spot. In a real experiment, you would not see any of this detail. You would only see the original microarray. Now we bound cDNA to see what is bound to the microarray. Let's detect the bound cDNA so it can be visualized. We begin by placing the microscope slide containing the microarray inside a scanner. We'll examine the next phase of the process, keeping our focus on the 3 spots we've been following. First, a green laser scans the microarray. The resulting image is stored on a computer for later analysis. Now it's time for the red laser. This image is also stored on a computer for later analysis. Now, we move to the analysis phase. After we eject and safely store the microscope slide, we retrieve the red and green images from the computer and create a merged visualization. In the merged image, we see an aerobic gene labeled in green, an red, and a gene labeled in yellow that was expressed in both aerobic and anaerobic conditions. This is one example of how DNA microarrays are used. In an actual experiment, quantitative analysis would be conducted on all 6,000 genes.
So for today, we might have another class now. It's 8 o'clock. So we will finish it here. So I will share with you this workshop of today. Make as much as notes you can. PCR, RT-PCR, DNA microarray. All these techniques we are done. Sir, computer science. बच्चे Sirjani saying if you want to do bioinformatics, then it is required, quite highly required. Then you can use this technique in that part. Otherwise, it's not. So okay then. If you have any other message, so I will keep sharing some MCQs with you in the day, and keep sending your assignments. Then I will keep looking them. The best one will be shared and will be appreciated. Okay then, ciao, take care, have a blast rest of the day. And today was Monday, yeah. So Monday went well, so the same way our whole week will also go well. All the best. Thank you. So videos are being shared within these presentations, so don't worry. See, the speed is so slow that it's not being uploading. Only 1%. Its speed is very slow today. Okay then, ciao, bye.